For today's webinar, we have Evie Nguyen, a hair examiner in the North Carolina State Crime Laboratory Trace Evidence section, presenting hair root staining. What can hematoxylin do for your laboratory? Evie will be joined during the Q&A by her NC State Laboratory colleagues, Lindsay Admire, Kristen Crawford, and Melanie Carson. Take it away, Evie.
Good afternoon, everyone, and thank you for taking the time to attend our presentation. My name is Evie Nguyen, and I'm a forensic scientist at the North Carolina State Crime Laboratory. Today, I'll be sharing the ongoing work of my fellow hair examiners, Lindsay, Melanie, Kristen, and I, regarding hair root staining with hematoxylin. First, I do need to provide our disclaimer statement. Forensic science is an ever-evolving and improving field. The North Carolina State Crime Lab updates its policies and procedures on a regular basis. Understanding that this presentation may be viewed at a later date, it is important to note that the information contained here may no longer be valid after a period of time. If you have questions or concerns about current policies and procedures, please contact the North Carolina State Crime Lab. Analysts cannot speak regarding cases that are currently being worked and/or answer other case-specific questions. If you have a case-specific question in relation to a case that the Crime Lab has worked, please contact Lab Legal Counsel Jason Kakamau at jkakamau@ncdoj.gov. All right, in our presentation today, I will first be covering some background information on hair as evidence, as well as the hair growth cycle as it pertains to suitability for DNA analysis. I will then be covering our initial assessment of historical data involving hair roots yielding DNA and casework prior to our lab's implementation of hematoxylin staining. Next, I'll provide background information on hair root staining techniques in general, leading into why we selected hematoxylin. I'll then cover the necessary materials for hematoxylin staining and give a visual overview of the method. Next, I'll go over the process of validating the method in our laboratory. We'll then review casework data involving hair root DNA yield after implementation of hematoxylin staining. Finally, I'll discuss some additional observations we had following the study. So why is hair evidence important? Hair may be left behind at scenes of a variety of crimes, including homicides, sexual assaults, breaking and enterings, hit and runs, and so on. Hair can provide valuable potential links between individuals, scenes, or objects, whether that be through microscopic comparisons or the topic of today's talk, DNA obtained from hair roots. Hair evidence becomes especially critical in situations where other evidence types may not be available. For example, say there was a breaking and entering case in which no DNA or latent evidence was able to be recovered, but hairs were found in the entry point that could be screened for any roots that may be suitable for DNA. Currently, sexual assault kit testing is a major discussion in the forensic field, and our laboratory is being inundated with kits to be tested, as I can imagine many of your labs are as well. In North Carolina specifically, sexual assault exams include hair evidence collection, and our lab's trace evidence section will perform testing on the sexual assault kits following any DNA analyses. Hair evidence is important to consider in sexual assault cases, as there are a number of reasons a DNA profile may not be obtained from the kit, such as a cleanup occurring or if the suspect were a condom. So in our laboratory, all kits first go to our lab's forensic biology section for DNA testing. If testing returns a DNA profile for each suspect, whether known, unknown, or even multiple, the case is considered complete and there is no hair analysis performed on the kit. This is the case either if the suspect is known with DNA standards available and the profiles returned are consistent with those standards, or if the suspect is unknown and the number of unknown DNA profiles returned and uploaded into CODIS is the same as the number of suspects. On the other hand, if DNA testing does not return a profile, either for none or just some of the individuals believed to be involved, or if for some reason the profiles developed are not CODIS eligible, the kit is considered DNA negative and will be transferred up to the trace evidence section for a hair analysis. Most often, this involves screening any human hairs present for roots that may be suitable for further DNA analysis. Items typically tested for hair include pubic hair combings, which are combings of the patient's pubic region for collection of any foreign hairs. In addition, underwear worn at the time of the assault are screened for the presence of any hairs, as well as any objects that may have come into contact with any transferred material from the assault, such as feminine hygiene products. If there is any indication that hair-like material was observed on any of the swabs typically included in the kit, such as the vaginal, oral, or rectal swabs, the hair examiner will examine that swab for the presence of a human hair as well. In order to obtain any potential DNA from a hair root, an analyst must first determine whether or not the roots may be suitable for DNA analysis. Historically, analysts would determine if any hairs present are human, if they have roots present, and then determine if the root had potential for DNA analysis based on the growth phase it was in. There are three main growth phases for hair roots: anagen, catagen, and telogen. The first phase is the anagen phase. This is the active growth phase of the hair cycle, in which the hair is firmly attached within the hair follicle and receiving an active blood supply. Hairs in this phase will not be naturally shed. It would need to be subjected to some amount of force for removal. The next growth phase is the transitional period between active growth and dormancy, known as the catagen phase. During this phase, the root begins to keratinize as it separates from the hair follicle, starting the transition from actively growing to resting. The previous elongated tissue in the anagen phase begins to harden into a bulb shape as the hair prepares to detach itself from the body. The final growth phase is the telogen phase, in which growth has ceased and any tissue adhering the hair root to the hair follicle is more fully keratinized into that more rounded, hardened bulb shape. There are two general types of telogen root we see in casework. Some with follicular tissue attached, as you can see in this photo, meaning it may be suitable for DNA analysis, and some without follicular tissue attached, as you can see in this photo, meaning it may not be suitable for DNA analysis. This image is an example of a hair that would be considered naturally shed, such as hairs that shed during typical grooming activities, such as washing or brushing your hair. These are usually the hairs found lying loose on your clothing or on your bathroom floor. Because of this, naturally shed hairs such as this are commonly encountered during forensic casework. At our laboratory, root suitability is determined by hair examiners in the trace evidence section. Any suitable roots are then sent onward to our lab's separate forensic biology section for the actual DNA analysis. Historically, we determined hair root DNA suitability based on the presence or absence of follicular tissue. Hairs with anagen and catagen roots are sent for DNA due to them being in some stage of active growth with viable tissue expected. Prior to the implementation of hematoxylin, telogen roots were selected for submission for DNA analysis based on the presence or absence of a follicular tag. For example, the image on the bottom left does have a tag present, so it is an example of a root that would have been sent for DNA. The image on the bottom right does not have a tag, so it is an example of a root that would not have been considered suitable for DNA. However, is presence of tissue alone enough for a hair root to be considered suitable? Despite the presence of follicular tissue, hair examiners noticed that hair roots being sent for DNA analysis were not yielding DNA profiles as expected. So, we decided to pull all of our hair root data over a 9-year period of casework from approximately 2008 to mid-2017. And looking at this internal data, we noticed 3 things. One was that telogen roots, as well as head hairs, are the most commonly encountered during casework, when root type or somatic origin was recorded. The second was although head hairs were most common, pubic hair roots tended to perform best when submitted for DNA analysis, likely due to their typically bulky tissues. And 3, only about a quarter of the telogen roots being sent were yielding profiles. So we did have a problem. Therefore, the hair examiners determined it was time to research methods to potentially improve DNA yields. The concept of assessing hair roots for the presence of tissue has been the generally accepted process of determining hair root DNA suitability within the hair community for the past 20 years. Meanwhile, forensic DNA technologies and capabilities have been ever-changing and expanding. Even with the shift in manual to automated analysis, as well as increased DNA sensitivities in our laboratory alone, we were still seeing the issue of a fraction of telogen roots returning DNA profiles. It was time for the hair discipline to catch up. Through reaching out to the community and review of the literature, we came across the concept of applying histologic stains to hair roots in order to visualize nuclei. Depending on the amount of nuclei present, thresholds could be set for a more objective determination of hair root DNA suitability, meaning only the best telogen candidates are sent forward for DNA testing. As DNA analysis consumes the root, it is especially crucial to preserve any non-viable roots for any future testing capabilities. At the time of our research, we came across these two articles outlining methods of hair root staining using DAPI and hematoxylin. So what is the difference in these two staining techniques? DAPI is a fluorescent staining technique that binds onto double-stranded DNA to allow visualization of bright blue ovular nuclei that may be present within tissue adhering to a hair root. Roots stained with DAPI must be observed under fluorescence microscopy. Hematoxylin is a widely used nuclear stain, common in applications of tissue staining within the biological sciences. Hematoxylin targets nuclear material within cells, revealing any nuclei present as dark red or purple ovals. Roots stained with hematoxylin are able to be observed under traditional transmitted light microscopy. And this is one of the main reasons we chose hematoxylin as a potential route, primarily due to its ease of use based upon the equipment and materials available in our laboratory. While the previous articles introduced DAPI and hematoxylin, an article by Edson et al. Modified the original Brooks technique for hematoxylin staining and outlined a validation study for implementation into the laboratory. We used this study as the inspiration for our validation. Here are the necessary materials, as well as an outline of the method provided by Edson et al. In the following slides, this method will be shown step by step. You should note this materials list includes equipment necessary for preparing roots for DNA analysis. This is the general setup for the root staining procedure. As you can see, the ethanol, hematoxylin, and deionized water are out and ready to go, along with a well plate, forceps, a pipette, and a glass microscope slide for the hair. To the right of the image, you can see sterile blades and a plastic microcentrifuge tube. These are materials you would need after staining is complete to cut and prepare a root for DNA analysis, if it has passed the nuclear threshold. Again, this is specific to our lab, where the trace section identifies, cuts, and prepares a hair root, and then sends it to the forensic biology section in the tube for DNA analysis. Before initiating a validation, we first needed to test the method in our laboratory. Pooled antigen roots were the first roots taken through the process to serve as a quality control step. If antigen roots are traditionally thought to have the most viable tissue present, numerous nuclei should theoretically be visible. The first step of the staining procedure is to soak the hair root in absolute ethanol for 30 minutes. As you can see in the image, ethanol has been placed into the well plate and the root is fully submerged. As this has to soak for 30 minutes and ethanol is volatile, it's important to cover the well before letting the root sit. We typically use a microscope slide for this purpose. Following the 30-minute ethanol soak, the hair root is then transferred into a new well and submerged with a few drops of hematoxylin stain. As you can see in the image on the right, just the root end of the hair is submerged, and the remaining shaft is not exposed to the stain. This is done in the interest of any future potential microscopic exams. During the study, we found that, especially with gray, bland, or damaged hairs, the hematoxylin may permeate the shaft of the hair as well. I'll show an example of this later in the presentation. So if possible, avoid staining the hair shaft. Once the 3 minutes of staining are up, the hair root is then transferred to a well of deionized water to remove any excess stain, and then into ethanol once again as a final rinsing step. Following the final rinsing steps, the hair is removed from the well plate and temporarily mounted on a microscope slide with either xylene or xylene substitute for viewing under the transmitted light microscope. The final step involves observing the root under the microscope to see if the stain has revealed any nuclei. They typically appear as dark red or purple ovals. So as you can see, the staining procedure worked and numerous nuclei were noted within the root, reinforcing the original hypothesis that antigen roots are believed to have the most viable tissue present. While specific to our laboratory, it is at this point in the method that a root would be cut from a hair shaft if it has been determined to have enough nuclei present to be DNA suitable. A sterile blade is used to cut as near the root as possible, and the root is placed into a plastic microcentrifuge tube for submission to our forensic biology section. The remaining shaft of the hair is preserved for future analysis if needed. But going back to our initial experimentation, now that we know the staining method works, how can we implement this method into our procedures? To prepare a sample set for the study, we solicited head hair donations from internal volunteers. We specifically requested naturally shed hairs rather than pulled in order to specifically target telogen roots. Participants were instructed to provide hairs from their hairbrushes or others naturally removed during washing or other grooming methods. Thousands of hairs were received between 15 donors and screened for the presence of any telogen roots. Over 900 telogen roots were identified and stained according to the method demonstrated previously. The roots were separated into categories based on the number of nuclei present. A sampling of roots without any nuclei present were utilized as the negative control. A small group of antigen and catagen roots from the sample set were used as a positive control. As shown in the figure, over half of the roots within the sample set did not have any nuclei present. This demonstrates the initial utility of root staining, narrowing down non-viable roots in order to preserve those hairs in case of any future analyses. At the same time, root staining highlights the smaller portion of roots with numerous nuclei present, allowing only the best DNA candidates to be sent forward. The following slides will provide visual examples of what roots within each nuclei group may look like. This is an example of a stained telogen root in which no nuclei are present. As you can see, this particular root has very little to no follicular tissue remaining, so this result is expected. Roots with this appearance made up our negative control group. It's important to note that as hematoxylin targets tissue, presence of tissue does not always indicate presence of nuclei. This is shown as the root has enough tissue present that stain uptake is visible, however no nuclei were noted. Oftentimes, telogen roots without follicular tissue will show very little, if any, dye uptake. Further examples of this will be shown later in the presentation. This is an example of a root in our group 1, roots with anywhere between 1 and 10 nuclei observed. This particular root has approximately 7 nuclei present, and they are all located within this small follicular tag at the top of the root. This image is a good example of how pigment and other particles within the root can sometimes be misleading, as well as the learning curve associated with recognizing nuclei under the microscope. While dark ovular shapes are observed towards the center of the root, these should be disregarded. Again, the nuclei we noted in this root are in this top tag. This root would be categorized into our group 2, roots with anywhere between 11 and 20 nuclei observed. This particular root has approximately 15 nuclei present, and they are again located at the top of the root within this small region of tissue. This image is a good example of a root that stained dark, nearing overstaining. It was observed during the study that a wide variety of factors may cause certain roots to take up a stain to greater degrees than others, even when all roots are stained for the same 3-minute increment of time. Reasons may include degree of tissue present, condition of the hair root, as well as slight variations in the 3-minute window between examiners. A recommendation for how to manage instances of overstaining or hard to discern nuclei will be provided later in the presentation. This is an example of a root with between 21 and 30 nuclei. which would fall within our Group 3 category. The nuclei on this root are located in the small region of tissue at the top of the root, as well as the mid-region of the root on the top surface. This is an example of a root in our Group 4 with anywhere between 31 and 40 nuclei observed. The nuclei on this root are located on each side of this large region of adhered tissue. And you may not be counting over 30 nuclei in this image. It's important to note many of the nuclei associated with this root are in a different focal plane than what is shown in this example. Nuclei are also located in the top center region of the root. Further discussion of focal plane adjustment and nuclei counting will be provided in the coming slides. This is an example of a root with numerous nuclei present that would fall into our Group 5, roots with greater than 41 nuclei. Here are a few more examples of roots in the 41 plus category. It's important to note that we've observed a wide variation of root appearance in general, which is most easily demonstrated by this nuclear category. For instance, big bulky tissue does not always mean nuclei will be present. Examples will be provided of this in later slides. Further, sometimes nuclei are observed in roots with large tissue, as best demonstrated in the top left image. However, numerous amounts of nuclei can also be observed in roots with not as much visually obvious surface tissue, as best demonstrated in the bottom left image. Also, as you can see, there is a variation in stain uptake color between the images. There are a variety of reasons these color changes can occur, including lighting conditions, degree of tissue present, camera settings, and stain lot number. And again, as anagen and catagen roots are in some stage of active growth upon removal, they are expected to have the most viable tissue and are therefore typically most successful in DNA analysis. Due to this and their commonly heavy presence with nuclei, our positive control group was composed of anagen and catagen roots. You may have noticed you aren't seeing the same amount of nuclei I'm pointing out in these images. It's important to note the limitation of attempting to portray a 3D root in a 2D image, and these images are highlighting the focal plane in which the nuclei are most easily viewed. Under the microscope, as you adjust your focus and change your focal plane, you will be able to observe nuclei as they appear in different regions and different depths of the root. However, take caution when adjusting your focal plane, as doing so may cause you to double count the same oval as you determine your total number of nuclei. In the video on screen, observe how the view of nuclei present at the top twist of tissue alters as the focus is adjusted. Examples of a circumstance in which focal adjustment should be done for most accurate nuclear counting will be shown in the next slide. In many stained roots, nuclei are observed in varying areas across the root surface that are not visible within the same focal plane. For example, in the image on screen, numerous nuclei are observed in the center region of the root. However, with an adjustment of focal plane, additional nuclei are now observed in the side regions of the root. Focal adjustment should only be done during nuclei counting in instances such as this, where nuclei are present in differing areas of the root. Now getting back to the validation, and now that our nuclear categories are established, the trace section began to collaborate with the forensic biology section, as again at our lab, the trace section screens hairs and prepares any suitable roots for DNA analysis. Then the roots are forwarded onward to the forensic biology section for the actual DNA testing. Based on the nuclei observed, a sampling of hair roots from each of these groups, including the negative and positive controls, were sent forward for DNA quantification. And as you can see, a clear delineation is observed between groups 1 and 2, with the percentage of telogen roots passing to DNA quant, where 36% of telogen roots with between 1 and 10 nuclei passed DNA quant cutoff, 80% of telogen roots with between 11 and 20 nuclei passed quant. At this stage, it appeared 11 or more nuclei may be the recommended nuclear threshold at our laboratory. A sampling of these roots that had passed quant were sent forward to amplification to determine if this delineation remains in the next step. Also, As we talk about DNA data specific to our laboratory, it's important to note these results will vary depending on your laboratory's DNA procedures, cutoffs, and capabilities. For example, the percentages of roots by nuclei number passing DNA quant may vary at your laboratory depending on your lab-specific DNA quant cutoff. Again, a sampling of the roots that had passed quant from each nuclear count category were sent forward to amplification. And according to the amplification data, the clear delineation remained between groups 1 and 2. Roots with 11 or more nuclei returned profiles at a much higher rate than roots with nuclei below that threshold. Specifically, telogen roots with between 11 and 20 nuclei were obtaining DNA profiles 89% of the time, while telogen roots with between 1 and 10 nuclei were only returning profiles 27% of the time. It should also be noted, our negative and positive controls did perform as expected, with none of our negative control roots returning profiles, which were roots with no nuclei present, and with 100% of our positive control, antigen or catechin growth phase roots, returning profiles. This data confirmed our hypothesis that roots with 10 or less nuclei should not be sent forward for DNA testing at our laboratory. These roots can be preserved for any potential future analyses, such as additional microscopic examination or other types of DNA testing that would not have been possible if the root had been consumed in analysis. The validation study also brought to our attention a potential reason our pre-hematoxylin DNA success rate was so low for telogen roots. As telogen roots were historically considered potentially DNA suitable with the presence of follicular tissue, the validation highlighted that DNA success is not so much dependent on presence of tissue, but on presence of nuclear material, as highlighted by staining. For example, the images on screen display a telogen root with follicular tissue both before and after hematoxylin staining. This root would have been sent for DNA analysis prior to hematoxylin, but as you can see, no nuclei are noted with hematoxylin staining. Our cutoff of 11 or more nuclei indicates this is a root that should not be sent forward for DNA analysis. Roots such as this example indicate why hematoxylin can be so useful for most efficient casework and best evidence practices in laboratories performing root screening. Once the validation was complete and our nuclear threshold was set, we implemented hematoxylin root staining into casework in March of 2019. Following implementation, the proportion of roots passing DNA quantification in casework was monitored for the next 2.5 years to establish an updated success rate. During this time, over 900 roots were stained in casework, of which nearly 500 possessed follicular tissue. According to pre-hematoxylin procedures, all of these roots would have been submitted for DNA analysis. However, with the application of hematoxylin staining, 70 of these 500 roots did not pass the nuclear threshold. This resulted in a 14% reduction in all hair roots being sent for DNA analysis, thus reducing laboratory time and resources. In addition, of the approximately 800 telogen roots stained in casework, approximately 200 passed nuclear threshold and were sent for DNA testing. Of these 200 telogen roots, a 65% DNA success rate was observed, compared to the pre-hematoxylin success rate of 27%. Some additional observations noted during the project included shaft staining, especially in gray, bland, or damaged hairs. This could affect any future microscopic analyses, so take caution to only stain the root end of a hair. An example of a stained shaft is provided on screen, located adjacent to an unstained shaft for reference. We've also noticed that hematoxylin oxidizes over time, especially when a bottle is nearing its expiration date. It may be useful to leave a root in the stain for approximately 4 or 5 minutes to achieve the same result. Further, due to this oxidation, the hematoxylin stain will oxidize over time, and small particles will accumulate within the stain that may transfer to the stained root, as shown in the image on screen. This oxidation is also noted by the manufacturer, and filtering of the stain is recommended to prevent these external particles. An additional consideration we took during the study was the potential effects of mounting media type on DNA results. 20 stained roots known to contain between 11 and 20 nuclei were mounted in temporary mounting medias, such as xylene or xylene substitute, and then sent for DNA analysis. An additional 20 stained roots with 11 to 20 nuclei were mounted in semi-permanent mounting medias, such as permount or cytosol, and then sent for DNA analysis. This was done to determine if any tissue loss occurs upon removal from a semi-permanent mounting media. While a slight decrease was noted from 80% in the temporarily mounted hair roots to 55% in the semi-permanently mounted hair roots, both DNA success rates are still above the pre-hematoxylin rate of 27%. This project was done as our lab performs hair comparisons prior to any DNA screening, as a means to narrow down the sample pool being considered for DNA suitability. During a hair comparison, a hair is mounted in a semi-permanent mounting medium. This data helped us to determine when it was best to stain the root, before or after the comparison. We chose to perform any staining after the comparison is already done, so the stain does not interfere with the microscopic comparison. This reduction in DNA success rate due to the semi-permanent mounting media is not observed in casework, as all staining is done after the comparison, once the hair has already been removed from the mounting medium. One of our most prominent observations following implementation of hematoxylin root staining into casework has been the decrease in roots passing DNA quant in our near threshold 11 to 20 nuclei category. Over the last 2.5 years of casework, telogen roots with between 11 and 20 nuclei have been passing DNA quant about 39% of the time. Although some decrease is to be expected in true casework samples that have been exposed to a wide array of environmental conditions and degradation as compared to pristine study samples. Nonetheless, we are actively monitoring root DNA performance in this category to determine if we should increase our nuclear threshold at any point in the future. Considering this slight decrease in DNA success in roots with near threshold levels of nuclei, it should also be noted stained casework samples have revealed a variety of appearances of potential nuclei. For example, some telogen roots have revealed the presence of bubble-like shapes with hematoxylin staining. These roots were sent for DNA analysis as it was unsure if these shapes were nuclei or not. These roots did not return any DNA results, so we now know to ignore any bubbles when observed on casework roots. Earlier in the presentation, I mentioned I would provide a solution for instances of overstaining, or if you're having trouble seeing nuclei. While one of the benefits of hematoxylin is that fluorescence microscopy is not required for its use, we have found fluorescence to be useful in situations of overstaining. There may be several factors causing overstaining of hair roots, such as slight variations in the 3-minute staining period or the condition of the root itself. We have observed that due to the wide variety of conditions true casework samples are received in, some hair roots appear to take up the stain at a higher degree than others. As you can see, the dark stain of the telogen root in the image obstructs your view of any present nuclei in transmitted light, but under fluorescence, as shown in this image, several nuclei are noted. Various types of fluorescence can also be used for this purpose. Here we are using an H3 filter, but as shown here, nuclei are also visible using a DAPI filter. This can be helpful in situations in which you are unsure if the shapes you are seeing are nuclei or not, as discussed in the last slide. For example, if you are debating if spots you are seeing within the root under transmitted light are pigment particles or true nuclei, you can view the root under fluorescence as the pigment particles will not fluoresce. Again, while not required, if your lab does have the capability for fluorescence microscopy, it can be a useful tool. As mentioned earlier in the presentation, often telogen roots without follicular tissue would barely take up the stain at all. As hematoxylin targets tissue present, this is to be expected. Telogen roots such as this were not sent for DNA analysis prior to hematoxylin due to their lack of tissue. As highlighted in our study sample set, over half of the stained telogen roots did not have any nuclei present at all, with roots with this appearance falling into this category. Therefore, this observation allows for analyst discretion in selection of which telogen roots to stain during casework. Telogen roots without follicular tissue were not considered DNA suitable historically, and hematoxylin staining has reinforced this. This observation is further supported by our data. Of the over 800 telogen roots that were stained over the last 2.5 years of casework, only 9 were noted that contained 11 or more nuclei that would not have been selected for DNA analysis prior to hematoxylin due to their lack of follicular tissue. And of those 9 roots, only 1 went on to pass DNA quant, indicating a 0.09% success rate of telogen roots with a visual absence of follicular tissue. In summary, DNA obtained from hair roots can provide valuable information to a criminal investigation. Prior to implementing hematoxylin root staining into casework at the North Carolina State Crime Laboratory, telogen hair roots submitted for DNA analysis returned results about 1 in every 4 attempts. A validation study of hematoxylin root staining within our laboratory supported a nuclear threshold of 11 or more nuclei. Again, it's important to note this validation is specific to our forensic biology section's DNA procedures. This threshold may vary depending on your laboratory's methodology. Since implementing hematoxylin root staining into casework, about 2 of every 3 telogen roots sent for DNA analysis are returning results. As hematoxylin is allowing us to be more selective in the roots sent for DNA analysis, this also means that less roots are being sent forward, thereby preserving more non-viable roots for future testing. So what can hematoxylin do for your laboratory? By decreasing the number of samples being sent for DNA analysis, it can increase casework efficiency, all while promoting best evidence practices through further evidence preservation. It can increase the success rates of the hair root samples that are sent forward, as only the best candidates are narrowed down as viable options. And it's important to mention that although our particular laboratory has a separate trace section that manages hair screening, this method is simple and straightforward enough that you do not have to be a hair examiner to carry out this process. We know it's becoming more and more common in the community that biologists within a DNA section are the examiners isolating hairs from evidence to be tested. Hematoxylin root staining is a simple method your DNA section could implement to narrow the amount of hairs being subjected to testing. All that would be necessary would be a modified quick training in macroscopic recognition of human versus animal hairs, presence or absence of a root, and the root growth phases, as well as hematoxylin root staining methodology. You don't even necessarily need a stereoscope, just the staining supplies and a traditional transmitted light microscope for nuclei counting. So now that we've taken you through our process, I'd like to show you a few examples of roots to highlight how nuclei present following staining may surprise you due to the initial appearance of the root. Here's my first example. Would you choose to stain this root? Feel free to type your responses into the chat. Due to the large amount of tissue present, I would stain this root in casework. Now, where would you expect to see nuclei once this root is stained? Likely in this large, elongated follicular tag present, right? Well, now that we've stained, do you see any nuclei? Let's move into a higher magnification. As you can see, when focusing on the elongated tag region, no nuclei are immediately visible. However, when focusing on the top region of the root, just adjacent to the elongated tag, nuclei are noted. This goes to show that nuclear material does not necessarily correspond with the most obvious region of tissue. Now, under the North Carolina State Crime Lab nuclear threshold of 11 or more nuclei, would you have cut this root for DNA analysis? How many nuclei do you see? I'll give it just a few seconds. I see approximately 12 nuclei here. So this is a near threshold root that I would cut and send for DNA analysis in casework. Here's an example of a root with bulky tissue present, which caused the analyst to choose to stain the root. However, do you see any nuclei within this root? How about in this example? These further reinforce the use of hematoxylin as a narrowing technique. So overall, there's a definite learning curve with hematoxylin staining as far as determining the appearance of true nuclei. And this should be kept in mind as you begin experimentation in your own laboratory. Here are the references to the 3 main articles we credit for the background information needed to complete this project in our lab. We'd also like to thank Faye Springer of the Sacramento County District Attorney's Crime Lab for sharing their lab's validation and helping us to get started with the project. We'd also like to thank Tricia Daniels of our Forensic Biology section for her participation in the DNA analysis side of the study, as well as our former intern, Samantha McDonald, for her work in the data collection. We are also appreciative to our crime lab colleagues for their donations of hair samples, as well as to our management for supporting this research. And with that, I'll open it up for any questions you may have. The 4 of us will be present to answer your questions live, but if you have any further questions, please feel free to reach out to our hair technical leader, Lindsay Admire, at her email address on screen.
Alrighty, thank you, Evie, for providing such an informative presentation. Our panelists will now answer any audience questions.
Hello, everyone. So one of the questions we first had was, does soaking the root in ethanol compromise the ability to get DNA since it's commonly used to decontaminate?
So from the research that we did prior to implementing, the ethanol soak actually is a good method for stopping more degradation of DNA that may be present in the cell. So we haven't seen any issues with the ethanol step compromising.
And what is the purpose of the second ethanol wash? And would it remove too much of the hematoxylin stain that could highlight any visible nuclei?
So the purpose of our second ethanol wash is essentially to remove any water off of the nuclei, or off of the root itself, so that when we put the hair root in the tube for DNA, we don't have the hair root sitting essentially in the water at the bottom of the tube. So the ethanol itself will evaporate and leave the hair root in a dry environment instead of a moist environment.
Okay, next question we have is there a way to visualize the hair root without mounting with xylene?
Unfortunately, you would have to mount the hair root in order to count the nuclei. But if you don't want to use xylene, you could use xylene substitute or deionized water. We generally prefer xylene or xylene substitute because the refractive index of those are closer to that of hair. So it's a little bit easier to see.
Another question is, how long did it take to actually carry out this study and implement the hematoxylin into casework? For that, this started as a summer intern project. It took about 2 years from doing the initial research, doing all the data mining, choosing the stain, performing the study, getting everything written into procedures to get this worked up and going in casework. Keeping in mind that the 4 of us do other casework, so we were full-time caseworking analysts while kind of handling this project on the side. And how often do we actually see the nuclei in casework?
So we've seen with our casework samples that they tend to follow pretty much the same distribution as our sample set during the study. The majority have no nuclei at all, or we're seeing roots in that lower nuclei category, the 11 to 20, or we're seeing roots with the 41 plus nuclei. So all the categories in between are kind of few and far between in casework, actually.
And question number, let's see, do other laboratories perform root staining that we know of?
We have heard of a couple other laboratories that are either performing staining or doing research on it, but we are always looking for ways to improve or discuss different options. So if you are a laboratory that's either doing the research or currently doing it, and you want to reach out, please feel free to do so. We would love to collaborate with you.
Great. Could the color of a hair play a role in the stain uptake? For instance, does light hair, such as blonde or gray, take up more stain than dark hair, such as black or brown hair? And if so, should the staining time be adjusted based on the hair color?
In the study itself, we didn't keep track of specific categories of pigmentation and color. Where we did notice some issues with the staining was more in hairs that were heavily damaged, especially along the cuticle area. So we would see that more in gray hairs or bleached hairs or heavily treated hairs, because the stain would almost infiltrate into the cuticle and within the cortex of the hair, and could cause darker staining to occur, which, if that did happen, was when we discovered potentially the use of fluorescence would help you to visualize the nuclei, because the pigmentation wouldn't necessarily fluoresce where the nuclei would.
Have we received any feedback from the biology section on whether or not the hematoxylin staining caused any PCR amplification inhibition?
Actually, we have received feedback from our biology section, and what we've been told so far is that it has not inhibited anything. And we're getting profiles, so we haven't had any issues come up. But if there is additional questions with that, feel free to reach out, and we can ask our biology section to give you a little bit more detail on that.
We were asked if we mounted the hair samples prior to staining with hematoxylin. In our laboratory, we do hair comparisons as well as screening for DNA. So in those hair comparison cases, we do mount using per mount. So prior to staining a hair, when we're removing that hair from a microscope slide, we are removing that per mount and any adhering residue before we initiate our staining procedure. We were asked, is this validation study being published?
So we are actually in the process of publishing the study, so that should be coming up sometime in the near future.
Okay, do we document the hair roots through photomicrographs prior to any of the staining that we do in casework?
We don't actually document any of the hair roots with photographs in our casework. The pictures that we took during the study were of our study samples and not of actual casework samples.
Okay. I think we kind of touched on this. Were any tests performed to determine if fluorescence microscopy has any adverse effect on the DNA yield? Strong UV light is known to denature DNA. We haven't seen any effects. We did some stability studies during our initial validation. For example, leaving some stained hairs on a microscope slide under transmitted light for several hours. We didn't see any decrease in results on our DNA yield with that. I don't think we've specifically studied the UV light, but again, we're not using fluorescence that often other than to visualize really darkly stained hair roots to see if we can see any of those nuclei.
And typically the exposure time when we're dealing with the UV light is typically less than a minute, I would say, that it's exposed. We just use it for visualization purposes, so we don't leave the hair exposed for long periods of time in the presence of the UV.
Is hematoxylin cost effective, and how much do we go through?
Gosh. We, unfortunately, the smallest you can order hematoxylin in is 4 pints. And we don't use that much of it. Even staining during the study, we didn't use that much of it. Each hair uses very little. So it is cost effective. It's unfortunate that we do waste quite a bit of it because it has a 1-year expiration. So when it expires, we have to put it in hazardous waste. So that's kind of the only drawback.
Thank you. If a root stains super lightly, are we able to restain that hair root?
So looking at the articles that we based our study off of, in the Edson study, they used a 3-minute staining period, which is what we typically do. But in the Brooks study, they actually did use a 5-minute period. So there is a little bit of a published range. So in casework, if we see that a hair has stained super lightly, we can put it back in that stain for a few additional minutes just to see if any additional nuclei are present.
Thank you. Do we have any concerns or have we seen any issues with hematoxylin in general?
As with most of the chemicals we use, it is carcinogenic. And also it is a dye. So if you drip it on your clothing, it will stain and it will not come out. And then of course, again, like I said before, we're, unfortunately wasting quite a bit of it. It would be nice if we could find a way to decrease that part.
Thank you, Kristen. Did we perform any stability studies during the validation? Touched on this just a minute ago. We did look at the ability to still visualize those nuclei once we've kept it on a microscope for a few hours using that transmitted light. We still were able to see all the nuclei from time 0 throughout that duration. We also, in starting to write up our paper and our results, and including for this presentation, went back and looked at hairs that we had initially stained, hair roots that we had stained back in 2018. And we were still able to see and count the same number of nuclei that we did back in 2018. So we've had a few years to study the stability there of the dye itself. With using hematoxylin staining, what is the increased analytical time per hair?
I would say that it has increased the amount of time we spend with the hair, because if we see any hairs that have tissue on them, then you're spending at least 30 minutes with the ethanol fixation step during that time, where previously, if we saw tissue, we could just cut the root that had tissue present. Now we actually have to add the fixation step, plus we have to add in the staining step, and then the visualization step, and the counting of nuclei present and whether or not it passes our threshold. So we have seen an increase in time on our end, but we decided essentially it was worth it because it means that we're getting better yield through DNA. So we would rather spend more time on our end before we cut the root and potentially don't get any information off of that root. And it could be something that maybe 5-10 years down the road, once science continues to advance, you could have gotten more information 5-10 years than you could today. So yes, it has increased our analytical time, but the increase in DNA that we've seen has been worth it.
We just have a couple more questions right now. If anyone has any follow-up questions, we'll be happy to answer them. We have a couple of DNA questions, specifically which DNA extraction kit was used. I don't know that we have the exact answer for that.
I believe we use the QIAGEN extraction process. So we do want to preface this by saying we're trace analysts. So specific DNA questions, feel free to send your questions to us, and we will forward them to the DNA section and get a response, and can get back to you regarding specific DNA analysis.
I see there was one more. Do you run a positive stain control with casework samples? We do always just indicate if we see that there is a dye color change, that purple reddish color appearing in our samples. Again, if we think that we need to restain or stain for a longer period of time, then we can do so in casework. So if there's any more questions, I'd be happy to answer. Well, if there are no further questions, we're going to go ahead and close out today's webinar. We want to thank you so much for being here today, and please feel free to reach out to any of us if you have any further questions. Thank you very much.
Before you leave today's webinar, please participate in our short 5-minute survey. Simply click the checkbox icon directly above the chat to access the audience feedback survey for today's event. Your feedback is critical information that helps us improve our webinars and continue providing the best experience possible. We would greatly appreciate it. Lastly, across the top of the live stream window are links to our social media profiles, including Twitter, LinkedIn, and Facebook.
There is also a link to the FTCOE website, the Just Science podcast, and the FTCOE newsletter registration. Like, follow, or subscribe to stay up to date with everything the FTCOE is doing in support of the forensic community. Thank you again for attending, and we hope everyone has a great day.